Today, an FDA approval in HER2-positive breast cancer, a priority review designation in lung cancer, a breakthrough therapy designation in head and neck cancer, applications accepted in ovarian cancer and breast cancer, and an ODAC decision in lung cancer. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved a supplemental new drug application for neratinib in combination with capecitabine for the treatment of adult patients with advanced or metastatic HER2-positive breast cancer who have received at least 2 prior anti-HER2-based regimens in the metastatic setting. The approval is based on findings from the Phase 3 NALA trial, which showed that the combination of neratinib and capecitabine reduced the risk of disease progression or death by 24% compared with lapatinib plus capecitabine. Moreover, the objective response rate was 32.8% for the neratinib arm and 26.7% for the lapatinib arm, respectively. The median duration of response was 8.5 months versus 5.6 months, respectively. Neratinib was initially approved for the extended adjuvant treatment of adult patients with early-stage HER2 overexpressed or amplified breast cancer following adjuvant trastuzumab-based therapy. In non-small cell lung cancer, the FDA granted a priority review designation to a supplemental new drug application for brigatinib for the first-line treatment of patients with ALK-positive metastatic disease, as detected by an FDA-approved test. The application is based on the Phase 3 ALTA-1L trial, in which treatment with brigatinib demonstrated a 57% reduction in the risk of disease progression or death versus crizotinib in patients with advanced ALK-positive NSCLC who had not received a prior ALK inhibitor. The 2-year investigator-assessed results also showed that brigatinib led to a 76% reduction in the risk of disease progression or death in newly diagnosed patients who had brain metastases at the time of enrollment. Moreover, with the 2-year follow-up data, the trial's results were evaluated and reported by both study investigators and a blinded independent review committee. At the data cutoff for the second interim analysis, results showed that by independent review assessment, there was a 51% reduction in the risk of disease progression or death with brigatinib over crizotinib. The FDA is expected to make a decision on the sNDA by June 23, 2020. The FDA has granted a breakthrough therapy designation to DEBIO-1143 for the treatment of patients with previously untreated, unresectable, locally advanced squamous cell carcinoma of the head and neck in combination with standard cisplatin-based concomitant fractionation chemoradiation therapy. The designation is based on results from a Phase 1-2 trial in which DEBIO-1143 plus CRT demonstrated a 20.8% improvement in investigator-assessed 18-month local regional control rate compared with CRT plus placebo. After a 2-year follow-up period, progression-free survival also favored the DEBIO-1143 arm. DEBIO-1143 was also found to have a predictable and manageable safety profile and did not compromise the full delivery of standard CRT. DEBIO-1143 is a potential first-in-class oral antagonist of inhibitor of apoptosis protein that sensitizes tumor cells to chemoradiation by promoting programmed cell death and fostering anti-tumor immunity. In ovarian cancer, the FDA accepted a supplemental new drug application for niraparib for use as a frontline maintenance treatment for women with advanced ovarian cancer who responded to platinum-based chemotherapy, regardless of biomarker status. The application is based on findings from the Phase 3 PRIMA study, in which frontline maintenance therapy with niraparib improved median progression-free survival by 5.6 months compared with placebo for patients with newly diagnosed advanced ovarian cancer who responded to platinum-based chemotherapy. In the overall study population, the median PFS in the niraparib arm was 13.8 months versus 8.2 months in the placebo group, translating to a 38% reduction in the risk of progression or death with the addition of the PARP inhibitor. In patients with tumors that tested positive for homologous recombination deficiency, the median PFS was 21.9 months with niraparib versus 10.4 months for placebo. Shortly after the FDA's acceptance, the European Medicines Agency validated a type 2 variation for niraparib as a first-line maintenance treatment for patients with advanced ovarian cancer who have responded to platinum-based chemotherapy regardless of biomarker status. In breast cancer, the FDA accepted a biologics license application for a fixed-dose combination of pertuzumab and trastuzumab with hyaluronidase administered by cutaneous injection in combination with intravenous chemotherapy for the treatment of eligible patients with HER2-positive disease. The application is based on findings from the Phase 3 Frederica study, in which the subcutaneous fixed-dose combination of pertuzumab and trastuzumab demonstrated non-inferiority to intravenous formulations of the two drugs with respect to pharmacokinetics, clinical activity, and safety. The total pathologic complete response in the breast and axilla and safety also were similar between the two treatment regimens. The safety analysis showed that incidences of the most common adverse events were similar between both arms, including alopecia, nausea, diarrhea, anemia, and asthenia. The FDA's Oncologic Drugs Advisory Committee voted 6-5 in favor of intravenous ramucirumab injection for use in combination with erlotinib as a frontline treatment for patients with metastatic non-small cell lung cancer whose tumors harbor EGFR exon 19 deletions or exon 21 substitution mutations. The FDA scheduled the Oncologic Drugs Advisory Committee hearing to discuss data supporting a supplemental biologics license application for the combination in this setting, which was based on findings from the Phase 3 Relay trial. In the study, results showed that the addition of ramucirumab to erlotinib led to a 41% reduction in the risk of disease progression or death compared with erlotinib alone in the first-line treatment of patients with EGFR-mutated NSCLC. The committee sought to discuss whether the risk-benefit profile of ramucirumab plus erlotinib could be adequately assessed without overall survival data, and whether the improvement seen in PFS is clinically meaningful in the context of additive toxicity. In the combination arm in Relay, the rate of grade 3 or higher adverse events was 72%, compared with 54% for erlotinib alone, and the rate of serious adverse events was 29% and 21%, respectively. This week, we sat down with Dr. Richard S. Finn from the University of California, Los Angeles to discuss the use of immunotherapy in patients with unresectable hepatocellular carcinoma.
In our management of unresectable liver cancer, we've really seen some dramatic changes in the past few years. For a decade or longer, all we had was sorafenib, which had shown its ability to improve survival in the first-line setting. And then for many, many years, we had numerous Phase 3 failures, both in the frontline setting as well as in the second-line setting. And in the past few years, we've seen the results of several Phase 3 studies, which have led to approvals of new drugs in the first-line setting, such as lenvatinib, and in the second-line setting, cabozantinib, regorafenib, and ramucirumab. There's been a big interest in immuno-oncology agents in all of cancer medicine, and this has eventually filtered down into the liver cancer space. Both nivolumab and pembrolizumab have received accelerated approval in the second-line setting based on Phase 1-2 studies. But the Phase 3 data for immune checkpoint agents, at least as single agents, has been difficult to get really strong positive Phase 3 data. For example, in the frontline setting, we saw at ESMO 2019 the results of Checkmate 459, which was an open-label study of nivolumab versus sorafenib. And this study came very close to meeting statistical significance, but it did not. And there was a lot of disappointment, I think, in that study. I mean, that study did confirm that nivolumab has a response rate in the 15 to 20% range, but we could not prove that it had better survival than sorafenib. In the second-line space, published in JCO this year, were the results of the Keynote 240 study, which was a placebo-controlled study of pembrolizumab in patients who had progressed on sorafenib. And this study actually showed survival benefit with the addition of pembrolizumab to best supportive care. However, when we look at the statistical design of that study, there were a few interim analyses as well as splitting of some of the alpha, the statistical power of the study between dual primary endpoints. And therefore, the study did not reach its statistical pre-specified significance. So there's been a big interest in IO in this space. That we know these drugs have response rates of single agents of 15 to 20% in both the frontline and second-line setting. We don't have a good biomarker to identify who those patients are that get a benefit. So the strategy has turned to combining these agents with other drugs to try to improve this response rate. There's been an interest now in combining IO agents with some of the tyrosine kinase inhibitors and also combining with bevacizumab, specifically atezolizumab and bevacizumab have been looked at in the IM-BRAVE 150 study. The goal has been to increase response rates to single-agent checkpoint inhibitors, whether that be PD-1 inhibitors or PD-L1 inhibitors. And there's been several strategies looked at. One strategy is combining these inhibitors with CTLA-4 inhibitors, and studies are being conducted looking at that approach, just like it's been done in other diseases. And then also, there's been data that has evolved over time that targeting VEGF does not just affect neoangiogenesis in tumors, but it does alter the microenvironment, that blocking either VEGF or the VEGF receptor can alter the T-cell subsets within the tumor microenvironment and prep a tumor for more of an immune response when combined with a PD-1 inhibitor.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.